In today's lecture at Medical Sciences, we want to discuss about the principles of determination of alkaline phosphatase, that is GGT and bilirubin. These are tests under liver function tests, and if you haven't watched about interpretation of liver function tests, please visit that video for more explanation of the interpretation of these liver enzymes and liver results. As we begin, if you haven't subscribed, please subscribe so that you're always notified and so that we can continue to grow together and share to many others. So I want to look at what is the principle behind determination of liver enzyme alkaline phosphatase, which is produced by the biliary tree, pregnancy and bones, GGT and bilirubin. And we want to begin with alkaline phosphatase. And this alkaline phosphatase enzyme, which we abbreviate as ALP, we use this method we are using is called King and Armstrong method. This is by King and Armstrong. King and Armstrong method. And the principle, here we are going to see this ALP. Remember, ALP is a hydrolase, or it catalyzes hydrolysis reaction. So we shall see that if we have a compound like P nitrophenyl phosphate, so this compound, which is known as P nitrophenyl phosphate, is going to be hydrolyzed by ALP. So we're going to see ALP enzyme hydrolyzes P nitrophenyl phosphate to form P nitrophenol. And this P nitrophenol formed is yellow in color. It is a yellow colored complex. Then we release the phosphate. So, and this, we are saying it is a hydrolysis reaction. This is catalyzing the hydrolysis of P nitrophenyl phosphate to P nitrophenol, which is a yellow compound, and this reaction occurs in the presence of at a pH of 10.5, or some books give between 10.3 to 10.5, in the presence of magnesium ion as a cofactor. So we are seeing ALP catalyzing the hydrolysis of P nitrophenyl phosphate in the presence of at a pH of 10.5 and magnesium ions to form P nitrophenol, which is a yellow colored complex. And we're going to see that this yellow colored complex formed is going to be monitored continuously. So the P nitrophenol, which is the yellow colored complex formed, is monitored continuously. So this is a kinetic method, is monitored continuously at 405 nanometer wavelength using spectrophotometer. You can use a spectrophotometer or colorimeter, but the best is spectro at 405 nanometer wavelength. And we shall end up saying that the intensity of the yellow color formed, that is P nitrophenol, is directly proportional. Is directly proportional to the amount of ALP in the serum sample. So this is how, So by the time a student tells us that ALP catalyzes the hydrolysis of this to form this, you get a mark. In which you get half a mark or a full mark, then these ones take half a mark. That is the conditions. Then you tell us that this one is monitored continuously at this is another mark. Then you tell us the intensity is directly proportional, another marker. So these are marking points. You see the marking points here. The marking points, these are the marking points. So don't write English, this is chemistry, so we have to be knowing which key terms have to be there. So this is the principle of ALP. The second we want to talk about is GGT, which is gamma glutamyl transferase. Gamma glutamyl transferase. This GGT is also by kinetic method, whereby we are going to see if we have arrow gamma glutamyl, arrow gamma glutamyl, and we add glycylglycine, and remember GGT is a peptidase, it is not a transferase. So what it does, it is going to transfer the gamma glutamyl group, transfers the gamma glutamyl group from gamma glutamyl nitroalanide, so it is going to remove this gamma glutamyl and he is going to transfer it to the glycylglycine, and it leaves nitroalanide alone. So at the long run, in the presence of GGT, we are going to form L gamma glutamyl glycylglycine, because we have transferred the L gamma glutamate group from nitroalanide to glycylglycine. So we shall remain with nitroalanide alone, plus nitroalanide. And what we are interested in is this nitroalanide formed. So we're going to see the rate of formation. We are going to monitor the rate of formation of nitroalanide, is going to be monitored continuously at 405 nanometer wavelength, spectrophotometrically, or you can use colorimetrically. And we shall see the intensity of this. We shall end up saying that the intensity of p-nitroalanide is directly proportional. To the amount of GGT in the serum sample. So this is where we end from, whereby we are looking, does the student know that the gamma glutamyl transferase catalyzes the transfer of this peptide group gamma glutamyl to glycylglycine, forming gamma glutamate glycylglycine, leaving nitroalanide. And we are going to monitor the rate of formation or the intensity of nitroalanide formed spectrophotometrically at 405 nanometer wavelength, and the intensity is directly proportional. So that is what we can talk about GGT. Then lastly is to talk about bilirubin, and this bilirubin contains both total and direct bilirubin. It contains indirect and direct. So this first one, we are going to see how do we determine total bilirubin. This is for total. And this method is known as Jedrasik and Groff. This is by Jedrasik and Groff method. That is how it is known. And this method utilizes nitrous acid, whereby if we have hydrochloric acid, it reacts with sodium nitrite, to form nitrous acid. And this nitrous acid which is formed is the one that is going to catalyze it is going to diazotize. That if we have sulfonylic acid, we are going to use sulfonylic acid is going to be diazotized by nitrous acid to form diazotized sulfonylic acid. This is what we form, and this one occurs in the presence of nitrous acid, whereby nitrous acid is the one that is diazotizing sulfonylic acid. So after getting this diazo reagent, which is diazotized sulfonylic acid, and this formation of this diazotized sulfonylic acid, we're going to see that if we have diazotized sulfonylic acid is going to be converted to pink azobilirubin. So this diazotized sulfonylic acid is the one that reacts with bilirubin in the sample. If there is bilirubin in the blood sample, this bilirubin reacts with a diazo reagent known as diazotized sulfonylic acid to form pink azobilirubin. So this is the pink azobilirubin which is formed, and this reaction is accelerated by caffeine benzoate. This one occurs in the presence of caffeine benzoate, which is acting as an accelerator. It is acting as a catalyst, acting as a catalyst. So if the asking objective is that which of the following is the role of caffeine during bilirubin formation, for it acts as accelerator, then the role of nitrous acid is to diazotize sulfonylic acid. Then after forming this pink colored azobilirubin, the pink colored azobilirubin is going to be converted, that if we have pink azobilirubin, it is going to be converted to blue-green colored complex. And how does it be formed? This reaction occurs in the presence of alkaline tartrate. So this alkaline tartrate is the one that catalyzes the conversion of this pink azobilirubin to form a blue-green, meaning this alkaline tartrate is oxidized, it is reducing bilirubin. It is reducing bilirubin to biliverdin, which is always a blue-green. So here we are forming biliverdin, which is a blue-green. And we are going to see this blue-green color which is formed, the blue-green color, biliverdin, which is formed due to presence of alkaline tartrate, is going to be measured colorimetrically. It is measured colorimetrically at 600 nanometer wavelength, and the intensity of this blue-green color is directly proportional to the amount of bilirubin in the sample. So here you have to tell us it is directly proportional, tell us the wavelength, tell us the function of alkaline tartrate, tell us the function. So in objectives, these are objectives. That during determination of bilirubin, which wavelength is used, or after reaction of a binkers of bilirubin with alkaline tartrate, which color is formed. So those are multiple choice. So here you can ask you more than 10 objectives in this principle. So whenever you are reading principles, don't only cram, but know which areas can we ask. Even here we can ask that during determination of ALP, which pH is used. I can put 10.5, 10, so something, then you have to choose. Or which of the following are used as a cofactor, that is magnesium ions. So after seeing this, I want you to note, if I want to determine only, that is NB, if I want to determine only bilirubin direct bilirubin, I will not react this with caffeine. I will do it in absence of caffeine. So when determining only direct bilirubin, it is done in absence of caffeine. Caffeine is not added. And after not adding caffeine, still you terminate the reaction. The reaction is terminated by adding ascorbic acid, that is vitamin C, which we are taking in COVID. So if you are interested in bilirubin total, both direct and indirect, here we are adding caffeine even to convert the indirect bilirubin. But if we are interested only in direct, we don't add caffeine as an accelerator, and then the reaction at a specific time is terminated by adding ascorbic acid. So this is what you have to master in case they ask you the principle of bilirubin direct. Thank you so much for learning with us at Medical Sciences. Please remember to subscribe and share to others. May God bless you.